So my group would like to replace neurons in the brain when they're lost or when they become dysfunctional due to Alzheimer's disease, old age, or any degenerative process. And we're particularly interested in one part of the brain, the neocortex, this outer folded tissue that covers just about every other part of the brain. It's the conscious part of our brain that we use to, for example, think, perceive, plan. So the neocortex defines us first as a species. It's very big. Up here in the left corner is the human neocortex, the outer part. And it's much larger relative to other species. And second, the neocortex defines us each individually because it holds our lifetime of memories and encodes our unique thought patterns. So other tissues we could simply replace with more functional tissues or whole organs, but we can't do that for the brain and certainly not the neocortex. That doesn't make sense. So next slide, please. Does it even make sense to replace the neurons of the neocortex, though? Could that change us each as individuals? Not necessarily. The neocortex is a very plastic tissue. And an example that I like to use is a study that compares individuals who have the same amount of damage due to either stroke or slow-growing benign gliomas. And the individuals who had the stroke, which is a sudden event, lose their ability to speak, whereas individuals, individuals who have the slow-growing benign gliomas can retain perfectly well their ability to speak even after the tumor is resected along with surrounding tissue for good measure. And if we look by functional MRI, we find that their language center has now moved to a different part of the neocortex, underscoring the plasticity of this tissue. Right? But this takes a long time to occur. So we want to replace neurons in the neocortex, but you may have heard we have billions and billions of these neurons. And in the neocortex, 15 billion alone, all organized in these in these nice layers. So this is a large number of neurons. So is it even feasible to consider replacing sufficient number of them to get functional rescue in cases of Alzheimer's, for example? Well, as a comparison, in the adult mouse dentate gyrus, 15,000 new neurons incorporate in 6 months. And if we scale that up for the volume of the human neocortex, that would mean that we could replace all the neurons of the neocortex within a year. Not that we want to replace them all, and certainly not that fast, but just to point out that the numbers in themselves are not insurmountable. I think a much bigger challenge is going to come from how these neurons are organized. Okay. All right, could I have the next slide? This doesn't seem to be working. Okay. So I showed you previously this picture with the human neocortex. The outside part is the neocortex. And down here is the mouse. Right? And they look nothing alike in shape or in size. But the mouse is actually the model system that we use. So how can that little thing tell us about the human neocortex? And if I asked you, what you thought the difference in thickness was between the human neocortex, the layers of the human neocortex, and the layers of the mouse cortex, what would you think? And most people are surprised to learn that they're very similar. Can I have the next slide, please? There's only a twofold difference in the thickness of the neocortex between humans and mice. And the layers are organized similarly in both species, in fact, in all mammalian species. So then, if that's roughly similar. Where does the difference come from? So next slide, please. The difference comes in the area. The mouse has about a square centimeter of neocortex flattened out, whereas the human, if you flatten the human neocortex out, it's about 10,000 times greater. And I think this is really where the problem lies in terms of thinking of cell replacement strategies for the neocortex. This is such a broad area that the production of new neurons would have to occur in a very dispersed manner. Next slide, please. So currently, there are different approaches, 3 different approaches at least, that are being used to address this problem. One is investigating whether there are already endogenous neural precursor cells in the neocortex that can generate new neurons. The second is, can we use other non-neuronal cells and maybe reprogram them if these cells are already present throughout the neocortex to give rise to dispersed neurons? And finally, can we engineer transplantable cells that will broadly disperse before differentiating into neurons? So the first one, next slide, are there already endogenous neural precursor cells in the neocortex that can generate new neurons? We and other labs have addressed this question, and the answer appears to be no. At least there's no precursors with an innate ability to do so to replace neurons after these are lost. Next slide, please. We use several transgenic approaches. 2 of them are illustrated here, where we can specifically induce the death of neocortical projection neurons. These are the neurons that are the principal neurons of the neocortex. And either with a diphtheria toxin or an inducible form of caspase 8, which causes apoptotic cell death. Next slide, please. We could induce scattered death of these neocortical projection neurons in the neocortex. And we looked at newborn neurons, next slide please, using the marker BRDU, and out of more than 25,000 newborn cells over many mice that we examined, we found no new neurons. And we used as a marker for new neurons co-labeling of BRDU with the marker NewM. So endogenous precursor cells do not innately replace neurons once they're lost in the neocortex. Maybe there are precursor cells there that we could potentially coax to become neurons in response to damage or loss of neurons due to Alzheimer's, for example. So another avenue of research that people are looking at is neurogenesis that takes place in another part of the brain, the dentate gyrus of the hippocampus. Next slide, please. So in the dentate gyrus of the hippocampus, this process of neurogenesis is understood at least at a superficial level. You have the stem cells in blue on the left here that progress to transit amplifying cells, then to neuroblasts, immature neurons and neurons. And there's a set of markers we can use to follow this process. Mechanistically, though, we don't quite understand what regulates this process. We have some clues, and the idea is that if we understand it better, then maybe we can apply those mechanisms to the neocortex and try to get generation of neurons in the neocortex to occur as well. So what we've done as an example is look at one family of secreted factors. Next slide, please. The FGF family. And we can specifically, using conditional genetic approaches, remove the receptors for FGFs from neural stem cells in the adult mouse brain. Or conversely, we can overexpress a form of FGF receptor that's constitutively active. So in one case, we get the loss of function, that's the LOF. In the other case, we get gain of function. And we can see what happens. Next slide, please. What we find is that FGF signaling is both necessary and sufficient for neurogenesis to occur in the adult hippocampus. So if we remove FGF signaling by removing the receptors, we get no more new neurons, or close to none. The new neurons are labeled as these orange dots, So in the control, you get a normal amount, and in the loss of function, you get essentially none. And if you increase FGF signaling, and this is the more exciting part, then you can get a considerable increase in the number of new neurons that are born and that incorporate into the adult hippocampus. Next slide, please. We also find that FGF signaling is necessary and sufficient for the rate at which, or at least increasing FGF signaling correlates with an accelerated maturation of the neurons in the hippocampus. So without FGF signaling, they mature very slowly, very poorly. If we increase FGF signaling and the gain of function, we see a greater number of branching of the dendrites, the projections of these neurons, and longer dendrites that occur sooner than they normally would. And next slide, please. And also as a molecular marker for a quicker rate of maturation of these cells is the co-labeling of both early and late markers, which normally hardly ever occurs. So DCX is an early marker, calbindin is a late marker. In the control, in the merged image, you see very little overlap. Whereas when you increase FGF signaling, you see lots of overlap reflected in the yellow projections of these neurons. So the neurons seem to be maturing faster as well. And interestingly, next slide, we find that FGF signaling also promotes the expression of phospho-Krebs, which is central to the process of learning and memory in the hippocampus. You can see that a lot more of the young neurons labeled in green express phospho-Krebs in red. Right, so that's also a very good sign that FGF signaling could potentially be used to treat Alzheimer's. Alzheimer's not only affects the neocortex, but actually affects the hippocampus first and more severely. So next slide, please. So FGF signaling can increase hippocampal neurogenesis, and it has some implications for treating diseases such as Alzheimer's. But it doesn't immediately address the problem of the neocortex, where you have this very large area in which you need to replace neurons, right? Maybe eventually we'll understand enough about how neurogenesis is regulated in the hippocampus to translate that to the neocortex. But right now we're still a ways away, so considering alternative approaches is worthwhile. So next slide, please. So the second potential approach that other groups are trying to adapt to the neocortex, which we're not using, so I'm just going to show you one slide on that to tell you what it's about, is to use non-neuronal cells and reprogram them into neurons. So next slide, please. So throughout the neocortex, there are cell types other than neurons, for example, astrocytes, microglia, vascular endothelial cells, and pericytes. And what this primarily one collaborative group in Munich between the Goetz and Berninger labs have shown, next slide, is that you can reprogram pericytes or astrocytes to become neurons, right? And this is potentially interesting in terms of cell replacement because these are already scattered throughout the brain. There may be a couple of caveats with this approach, such as astrocytes and pericytes are probably already doing something very important in the brain, and if you reprogram to become neurons, you might get other problems. The second issue is targeting these cell types for reprogramming specifically throughout the brain. So you may have the same sort of conceptual problem of reaching cells in that wide area. So what we're doing is taking an alternative approach. Next slide, please. And that's trying to engineer cells that we can transplant. And these cells would disperse widely before differentiating into neurons. Next slide, please. So if we just take embryonic cortical precursor cells, the cells that normally give rise to the neurons of the neocortex, transplant them in the adult, they stay in these clumps at the transplant site. They don't disperse. During development, they don't have to disperse. So they're essentially doing what they normally do. Other groups have tried other types of neural precursor cells, adult neural stem cells. They do the same thing. They stay in these clumps. There is one type of neural precursor cell, however, that loves to disperse in the adult neocortex. Next slide, please. And these are MGE precursors. MGE just stands for medial ganglionic eminence. It's a transient structure during brain development that gives rise to interneurons that migrate long distances. So when we transplant these cells in the adult neocortex, they disperse. And the next slide just shows the quantitation of that. So this is distance migrated from the transplant site. In blue are the cells derived from these MG precursors, so they're dispersed for long distances compared to the cortical precursors that I showed you stay in a clump. They essentially don't disperse at all. But of course, these cells give rise to interneurons, not the neocortical projection neurons that we want to replace. So the strategy we want to use, next slide please, is to collect the MGE from these early embryonic brains, dissociate the cells, infect them with lentiviruses carrying candidate reprogramming factors, then stereotaxically inject these cells into the adult neocortex in an Alzheimer's disease model or just for proof of concept in wild-type mouse. Wait for the cells to disperse, then hit them with a factor, in this case doxycycline, that induces the expression of the lentivirus-encoded candidate transcription factors, and then wait for these cells to differentiate and see whether or not they give rise to cortical projection neurons or to the interneurons, which we don't want as much. Next slide, please. And so here again is the DNA constructs that we use. So the donor cells carry two transgenes. One carries this RTTA transcription factor. That's simply to allow us to induce, next slide please, transcription of the virus. So of the candidate transcription factors, and I'll tell you what those are in a minute. And also to induce the DNA recombinase CRE, next slide please, which then recombines another transgene in the donor cells to permanently label these cells throughout the life of the animal with GFP in their membrane and LAC-Z, or the protein that encodes beta-gal, in their nucleus. So with this, we can follow the cells after transplant for the life of the animal. Next slide, please. These are the transcription factors that we're using. They're all selected because during development, they specify neocortical cell fate as opposed to an interneuron fate. And this list is actually longer now. We have about 25 factors that we're testing. Next slide, please. And every step of this approach is working so far. So we can infect large number of MGE cells with these lentiviruses. Over 50% is a standard. We can induce expression of the candidate transcription reprogramming factors with doxycycline. An example here is shown with the transcription factor NGN2. So the cells that are infected are in green, and we induce expression of this transcription factor specifically in those cells upon administration of doxycycline. Next slide, please. And we can follow these cells in the animal forever, well, indefinitely, because as you see, their membranes are labeled in green, so you can see their projections very nicely. You can also see their nuclei, which is very convenient for counting them, labeled in red. It looks kind of orange here. And finally, next slide, please. We can reprogram these cells, although our efficiency right now is very low. So about 1 in 900 of the infected cells is reprogrammed from an interneuron fate to a projection neuron fate, which is what we want. And we use as a marker TBR1 because it's specific to projection neurons. The efficiency is low because we're still working with pools of these candidate transcription factors. We have to narrow down which factors in those pools are actually responsible for the reprogramming, and then combine those essential factors and hopefully get a high efficiency. Others now, using chromatin remodeling factors as well, have upped reprogramming efficiencies to close to 100%. We'd be happy with 50%. We're also including reprogramming factors now in our list of factors to reprogram. So we're hoping to get the efficiency much higher. But we're also, in the meantime, as we're doing these transplants, learning some pretty interesting things. Next slide, please. One is that if we transplant these cells in an environment that's undergoing neocortical cell death, such as in Alzheimer's, the transplanted cells will actually migrate even further, which is a good thing. So in light blue are the cells transplanted in a normal, healthy adult environment. And the darker blue are transplants of these precursor cells in an environment undergoing cell death. So they don't mind that there's cell death. In fact, they seem to migrate a little further. Next slide, please. And as a control for proof of concept, we're also injecting not just these MG precursors that disperse, but cortical precursors, which I showed you earlier, don't disperse. And again, here's a rather big transplant site. Every red dot you see here is a transplanted neuron. Next slide, please. If we zoom in, it's a little hard to see the box, to the edge. Next slide, please. You can see that it's a very well delineated transplant site. So these cortical precursors, unlike the MG precursors, as I mentioned, don't disperse. They do differentiate, however. And so we're still curious to know, well, what do they do in the adult environment? Next slide, please. Again, next slide. So one of the things that they do is survive. Next slide, please. And that's a bit surprising. Even in the center of the transplant, the cells seem very healthy, even a month after transplant, which suggests that they must be getting nutrients from somewhere. And in the transplant site, it's mostly the neurons that are derived from the precursor cells and not much else, but there must be some blood vessels. Next slide, please. So we looked with a marker for blood vessels, PCAM. You can see, surrounding, this is a smaller transplant site, and the transplanted cells are in green. You can see PCAM labeling blood vessels surrounding the transplant site, but also within the transplant site. So somehow, these transplants are inducing the formation of new blood vessels. We don't know if those are derived from host cells or from transplanted cells, because we don't purify our precursor cells before transplanting them. But either way, it's interesting that they have the potential to organize blood vessels and maintain their survival for long periods of time. That's an important aspect when we're thinking of regeneration. Next slide, please. We also can generate specific subtypes of cortical projection neurons. I showed you the neocortex is arranged in these layers. And with these transplants, we're generating upper layer neurons. So you can see on the left here in red, you can see this track. Those are the endogenous host upper layer neurons labeled with this marker COOX1. And you can see the transplanted cells in green here labeled with beta-gal. They also express COOX1. So we're generating upper layer neurons, even though they're in the deep layer. We have to adjust where we actually transplant them, but we can generate subtypes of layer-specific neurons. And you can see that in the overlapped, the merged image as well. Next slide, please. But maybe one of the more exciting findings is that these neurons can project axons for long distances to normal targets. So the little cartoon on the left here shows you a cross-section through the mouse brain. The two top parts are the neocortex, and the little red pin is where the injection site took place. And then the 4 green boxes are the enlargements here. So you can see right next to the transplant site, all the green in that slide, those are all projections coming from those cells. Right, so very exuberant projections in the neighborhood of the transplant, which is what would normally happen. The cortical projection neurons project exuberantly in their local environment. We also see projections that go to the contralateral hemisphere. In this bottom left panel here, you can sort of see where the midline is, the transplant's on this side, and you see all these tracks here heading across to the other hemisphere. Right, so those are going long distances, sometimes up to 2 millimeters in the mouse brain. And finally, they project to subcortical targets such as the striatum, again in the bottom right there. So this is encouraging. This tells us that if we can get these MGE cells to disperse before differentiating into these cortical precursor cells, that they also should be able to functionally incorporate. And now we're taking this project to the next step of using human cells. We can get human MGE cells, either from ES cells or IPS cells. Those protocols are already established. And use those to test first in mice, then primates, and then, hopefully in the not too distant future, take those to trial. So with that, I'd just like to thank those who've actually done the work in the lab. Next slide, please. So in bold are the ones that have contributed in one way or another to this project. Helen, Pia, Martha, Wente, Joanna, Nicholas, Frank, and you, and the funding agencies. So thank you, and if you have any questions.